And I'm very glad to introduce our speaker today, Dr. Emma Robinson. Emma is a professor of psychopharmacology at the School of Physiology, Pharmacology and Neuroscience at the University of Bristol, UK. She completed her undergraduate studies and PhD at the same university. And after working with Professor David Nutt in the psychopharmacology at the University of Bristol for several years, she continued the fellowship at the University of Cambridge with Trevor Robbins and Jeffrey Daly, and then returned to Bristol to establish her independent research group, where they are using behavioral studies alongside with neuropharmacological and neurochemical approaches to study the role of specific neural and neurochemical systems in the control of behavior. And they are particularly interested in developing novel models that can be used in humans also to study the cause and treatment of psychiatric conditions where emotional changes are an important feature, like depression and anxiety. Importantly, I learned also that Emma has been identified as the most engaging and inspiring lecturer by the students at the Bristol University. So we are looking very much forward to her talk, a critical review on conventional models of depression and alternative approaches to studying emotional behavior in non-human species. So please, Emma, screen is yours.
Thank you very much for that introduction. I hope I live up to what my students said about me. Anyway, so what I wanted to do, I thought I'd give you a bit of my perspective around conventional models of depression and then some of the alternatives that we've been developing and talk you through kind of the process we went through in developing those. And so a lot of what I'm going to talk about is going to be this task, the forced swim test, which I'm sure you're all aware of, has been discussed a huge amount recently in the literature. And then I'm going to introduce this affective bias test, which was developed in my lab. And I think has, I mean, it's taken us about 10, 15 years of development. So I think it demonstrates that it's not an easy thing to develop new behavioral readouts. But I hope I'll be able to show you how we've kind of gone through the process of trying to really understand whether or not this is achieves the validity we need to study emotional behavior related to depression in non-human species. So I should just, my disclosures, so I do some consultancy work for Compass Pathways and Pangaea Botanica, and then we have current grants where we have collaborations, or I do some CRO work for a number of different companies. So I thought I'd just start with a little bit of context from my point of view, really, and that is, you know, why is it that we seem to have found it so difficult to understand and treat disorders like major depressive disorder? And I think that we sometimes forget that actually we still have a very limited understanding of the causes of diseases like major depressive disorder. And that's because they're really challenging disorders to study, not just about the animal side of it, which I'll focus on in a minute, but just from the clinical perspective. They're diagnosed with subjective self-report type measures. We don't have biomarkers. We have a lot of variability, heterogeneity in the populations. And we have limited methodologies. It's much more difficult to study the human brain, as we all know. Interestingly, we also still actually have a surprisingly limited understanding of how the drugs work. And I think, you know, I like to think of it as playing catch up. Some of you may be familiar that drugs like imipramine, our first antidepressant, and true of all our psychiatric disorders, that these were discovered serendipitously. So these were drugs that were being used for something else. Observations led to them being recognized as affecting mood. They were then used in patients with primarily a mood disorder, found to have efficacy, and that's then led to their use. But the drugs worked, but we didn't know how they worked. And then we've kind of been understanding more about their pharmacology, but we still don't really know what it is about giving a person an SSRI, for example, and why their mood actually gets better. We don't have that link between the kind of biology and the behavior. And actually we see all variable clinical benefits. We don't really know why is it that some people respond and some people don't. And there's been this really interesting, when I started my sort of research career as a PhD student, it was all about delayed onset of antidepressants, and at that time everyone just accepted that antidepressants took weeks to have their effect. And then, you know, in 2000 ketamine came along, and showed this amazing rapid acting antidepressant effect, but it kind of threw everything in the air, because suddenly we had a drug that was working quickly. It had this sustained effect. So it was a real breakthrough, but then it presented new challenges. How do we kind of synthesize this to understanding the underlying mechanisms? And I think the real challenge, and I think I'm not the only one, lots of people have recognized that we have real limitations around our current readouts used to with animal models of depression. So I'm not really focusing here on the phenotypes, the things that we do to induce depression. Most of our animal models are based around sort of stress-induced deficits, but really it's about the readouts and how do we quantify that depression-like behavior. And essentially all the readouts that existed when I certainly started my career, and the vast majority that you'll see in the published literature, they don't really recapitulate the human condition. These are pharmacological screens, really, that we've subsequently then tried to use, probably because we sort of didn't have anything else. And the real issue is that there's lots of examples of false positive and false negatives in these tasks and in these behavioral readouts. And that means that people lose confidence in what the data will mean. And it also means that the underlying mechanisms may not involve the same mechanisms as are going on in the human condition, and therefore how that drug might even be working in the human condition. So I thought I'd just sort of highlight, I mean, I've got a few rather texty slides to begin with, but it was really just because I wanted to kind of pitch my perspective on this field and where I see we are at the moment. And we've got these sort of requirements. We all kind of know what we want to see in an animal model for depression research, but I've kind of tried to highlight why this is not quite so easy as people say to you, well, you need a better animal model. It's like, well, we know that, but actually these are really difficult disorders. So we would obviously want to model the underlying pathology, but if I ask any of my psychiatry colleagues, they tell me we don't know the underlying biology. So I'm a basic scientist. I'm not a clinician, but if I talk to clinical colleagues, they don't know. We know about risk factors. We know about there is some genetic components. We know about environment, but what we don't really have a handle on is how is it that those things lead to some change in the brain? What is it that changes that then leads to the symptoms that that person experiences? You want your model to show equivalent changes in biomarkers. And so the classic, blood pressure or insulin levels, these are things that we can measure in an animal, we can measure in a human, we can test a drug, we can see that drug changes, or we can look at a mechanism. So we can, look at, for example, making an animal obese, start to induce obesity, and we can see the insulin resistance develop. And so it's a biomarker. So it's something that we can measure and quantify. But we don't have that for major depressive disorder. There's been a lot of discussion around BDNF, but to be honest, blood BDNF is certainly, from what my understanding of the literature is, it's really not that reliable as a marker. And it certainly isn't specific to major depressive disorder. It's changed in many psychiatric disorders. So we don't have a biomarker. We do need it also to respond to clinically relevant treatments. There's a lot of controversy, yes, still about antidepressants, but people generally, the consensus, all the metadata says that antidepressants do benefit patients, particularly in preventing relapse. And in the longer term, we get big placebo responses, but then in the longer term, the placebos tend to wane, whereas the antidepressants continue. And that's where the forced swim test really was developed. And actually, there is still some validity in the forced swim test being used, but only for very specific drug classes where we know that its efficacy in that test for monoaminergic reuptake inhibitors, for example, that's got a reasonably good translation to clinical efficacy. But for all the other targets and potentially sort of novel targets, it's a bit of an unknown quantity. And we also want to recapitulate relevant symptom domains. And so this is really what I got interested in. And I started to look beyond the subjective self-report measures and started to say, well, what is it that happens in people with these disorders that we might be able to measure in animals? So we know there's not a blood biomarker. We know that we're not really going to be able to easily get a handle on pathology. The pathology is not a gross, there's not like stroke where you can see an obvious underlying pathology. But if we went to neuropsychology, we could see some really interesting things. So objectively measuring how people process information, their cognition, it started to reveal, and that literature has been growing quite nicely over the last 20 years. And in fact, it was back in the 1960s that talked about this kind of very much more cognitive model of mood disorders. And these things, two areas that we were interested, affective biases and reward learning impairments. So these are very much around cognition. And I'll talk a bit more about those in a minute. And that's sort of how we've tried to look at it. So I'm going to talk about the forced swim test because I was asked to sort of provide a bit of a kind of critical appraisal, I guess, of the conventional models. And there's been so much in the literature recently about these. So I don't know how much familiarity people have. So apologies for those who use this test and know this test well, but just as a sort of an overview. So the forced swim test, this was originally developed by Persault, and it was designed as a predictive pharmacological readout. So the aim of the task was that it would predict drugs that were similar to imipramine, amitriptyline, ipronizid, these reuptake inhibitors, monoamine oxidase inhibitors, which had been found to be antidepressant in people. And although there is a lot of criticism, and some of that is probably justified, I think it's also really important to remember that this test has been critical in this development of second generation antidepressants, which are some of the most widely prescribed drugs in the world. And it came about because the test could identify drugs with similar pharmacology, and because they had this in vivo effect, it could then predict whether or not it was worth taking forward to clinical trial. Where I think it started to go wrong was at that era when we all got very excited about the kind of molecular genetic side of things and genetic risk factors, and we had GWAS studies delivering us these potential genes of interest that we then wanted to mutate in animals and look at the phenotype. And also because, as industry had started to sort of point out, we've got our Me Too drugs, we still haven't made a huge progress in efficacy. We need new drugs. We want something new. And when we started to think about new, we wanted to understand the mechanisms of depression. And this is where I think it's gone wrong. So I think using the forced swim test around considering whether you've developed a depression-like phenotype and also the underlying mechanisms, including, I would argue, the underlying mechanisms of our conventional antidepressants. So the problem is it lacks construct validity, and that's where we've seen this really poor translation of outcomes. So the translation of the findings in animal models has really failed to translate. There's a sort of a long history now of drug company failures with antidepressants that did look positive in the forced swim test, but have then subsequently failed. So rimonabant's a great example. It's a drug that was developed as an anti-obesity drug. It actually looked antidepressant in the forced swim test, but then it was withdrawn because of causing suicidal ideation and behavior in people. So really it demonstrated that the assay for the cannabinoid pharmacology was wrong. It couldn't predict. The concerns primarily relate to the validity of the task and its implications for much of the kind of preclinical depression research. So this is about how do we advance our understanding of underlying mechanisms of emotional behavior, identify new drug targets, and that's where I think the forced swim test has certainly got issues. But the problem is there's a lack of alternatives, and I think that what we widely do is there's a sort of nothing. We've got nothing else, so it's a better than nothing attitude. We don't have good alternatives, and the forced swim test is really, to a lot of people, it looks very easy. It's a short term, low equipment, that kind of thing. Interestingly, it's also been the focus of a campaign by an international animal rights organization, which many of you may be familiar with. Bristol's been a target for that organization, but they've been targeting around the world and targeting the Home Office around the use of the forced swims, and I'm calling for it to be banned as a test. So there's a number of articles here. So this is the Persault paper, the original Persault paper, and then you've got I should put my laser pointer on, hold on, and then you've got the articles recently in places like Nature and also other journals really calling for a rethink of how we model depression. I don't know my slide, let's go, there we go. So from a current sort of my views on the current perspective, so there's a lack of kind of valid readouts for establishing the relevance to human mood disorders and emotional regulation. We've got some others, so I haven't put lots of information in about these. I want to focus more on kind of some of the newer ideas and the approach we've taken, but you'll be probably familiar with the sucrose preference test and the novelty suppressed feeding test. These are more models around reward sensitivity, anxiety, but they do suffer similar criticism. So difficulty with understanding how they translate to human symptoms, poor understanding of their underlying mechanisms, tend to work quite well in chronic stress models, but not in other models, and therefore perhaps not really tapping into those core mechanistic pathways that are important for depression. The pharmacological effects in the forced swim test are actually not reliable readouts beyond these very specific classes of monoaminergic drugs. And in fact, there's some people have done these kind of big analyses and shown that, you know, almost every CNS drug at some dose will have an effect on behavior in the forced swim test. And so it's really important that when using the test, people recognize that and understand the limitations of it and use it appropriately. And I think that's probably where a lot of the issues have come from, is where the test hasn't been necessarily used in the best way, or perhaps has been used but then interpreted to mean more than the data would actually support. So yes, you might have seen a change in immobility time, but maybe the drug was given at a really high dose, and therefore it's not really engaging the right receptor population. Interestingly, as far as I know, the mechanisms underlying this immobility time, which is the kind of key measure, and how it's then modulated by treatment, is not known. So we don't know, and I think this is the difficulty with animal models in the CNS in general. But just because the animal's got a behavior and that behavior has changed, do we know that it's involving the same underlying mechanisms as those that are relevant to human depression? So it only really shows reliability for phenotypic studies where, well, not even reliability, consistency is probably the better word, which are associated with chronic stress. So there's reasonably good consensus data now that if you chronically expose an animal to stress, so quite severe stressors, long repeated restraint stress, chronic mild stress in the home cage, chronic treatment with corticosteroid, that will pretty reliably impair behaviors in these conventional methods of models of depression. But we know there's other risk factors for developing depression, and they're not so reliable. So things like early life adversity, and we've looked quite a bit at an immune mediated model, and there you just don't see these consistent changes, which is quite interesting, and suggests that perhaps this is about stress and stress coping. And we know stress is really important in psychiatric disorders, so I think there is a place, but how we interpret it, I think we've got to be careful not to overinterpret that as directly linking to depression. So although it's a fairly simple test, the forced swim test involves, it's an acute test. Animals are placed into an inescapable beaker of water, and their time to immobility is recorded. Mice, it's generally done as a single test. In rats, you have a kind of pre-test, and Paul Salt designed it with a pre-test and then 24 hours later. So there may be an element of learning in it. But it's actually, although it's relatively simple, it's actually been found to be surprisingly variable, and with lots of factors that influence the behavior in the forced swim test. And this has led to a lot of inconsistencies across the literature. And then again, it raises challenges around the interpretation. So I think that the Catherine Commons papers are really interesting and nice review of what it is and isn't looking at. And I think that's the one we use a lot for reference in terms of what the forced swim test. It's around this kind of stress coping, and stress coping in itself is a really interesting behavior. You get animals who cope, animals who are resilient, and I think there's some really interesting questions that we can use it for there. But what the conclusion that they drew was that it's not depression-like behavior. We can't directly then interpret that in relation to human depression. And there's a couple of other big reviews here about the different factors that sort of influence it. So particularly methodological considerations and its relationship to other behaviors. And certainly, I think it has a place still in some studies where you might want to look at it but in a battery of behaviors, so that it's not just a single readout. You can understand how it changes relative to different behaviors. And then you've also got factors influencing the behavior in this review, which is things, water temperature, sex differences, strain differences. There is quite a lot of variability in the task itself, and that again makes it more challenging to use. And certainly my work with industry have pretty much lost interest in using this. They don't see it as a particularly good assay in the whole drug development sort of process. So let me sort of move on from the kind of general background to sort of talking a little bit more specifically about the way that we approach this, and particularly the challenges. And I was very aware as a PhD student when I started using I was doing a project looking at whether we could use a genetic manipulation to quickly knock down the autoreceptors in the brain and generate an antidepressant-like outcome. And I was a bit surprised as a naive PhD student that when it came to the readout, I was going to look at this forced swim test, and I just didn't really understand how it aligned with. And then people started telling me, well, it's not really. We don't really know what it is and what it does. And I remain sort of rather dissatisfied that we didn't really have a model, a readout that I thought was, and so I was kind of keen. That was always what I wanted to do, was try to make a better model. Of course, then you come up with lots of interesting challenges. So you realize that depression is almost entirely quantified in humans based on subjective self-report measures, and you know, we work a lot with animals, but there's no way that we can really relate these unless we apply some sort of anthropomorphism, and that's where the problem is. It's a human interpretation of an animal's behavior against what we think happened, what we know humans self-report. But actually that's really difficult. So the kind of animal models that people have mostly used, and these are the ones most widely used, are sucrose preference tests. So do the animals like and prefer to drink a sweet solution is linked to this symptom of loss of interest in rewarding activities. The behavioral despair that we see in the forced swim test has been linked to this construct of hopelessness. And then the novelty suppressed feeding is linked to this kind of idea of worry. But these are very subjective interpretations, and we don't know what an animal's experience of hopelessness is, and we don't even really know what loss of interest means in terms of the human versus, you know, does this relate to an animal drinking a sweet solution? And so there are some huge challenges in terms of how do we relate these kind of symptom domains to what we can measure in animals. And I've always argued that I think it's both a clinical and a preclinical problem. It's not just a preclinical problem. We need clinicians, we need measures in humans that we can translate to animals, because unless we have that data in the human literature, then it's very hard for us ever to validate our animal models. So this is the area we got interested in, and this is this idea of affective biases in depression. And I didn't really know much about psychology until I went to work with Trevor Robbins in Cambridge, where I started to understand a little bit more psychology. I'm definitely not a psychologist, but I realized that this was possibly the best route we had to something objective that we could measure in humans and animals. And I think, and I've been pushing this idea that perhaps rather than trying to find biological, sort of the biochemical biomarkers, blood biomarkers, perhaps we should recognize more that psychiatric disorders by their nature actually influence because they influence cognition, that really what we should be interested in and what we should be trying to categorize are cognitive biomarkers. And if we have cognitive biomarkers that we objectively can measure, then we can measure those potentially in different species. Now, affective biases are really a kind of interesting thing. We've all experienced them, and they are when our emotional state biases the way we think, the way we interact with our environment, and how we and our executive functions. And so these are simply things, you know, we've all experienced it. If you're walking down the street late at night and it's dark, you're going to be more sensitive to noises and potentially threat, and that's because you're in a slightly anxious state because it's dark and the environment is anxiety generally. That's completely normal. Those biases have huge advantages to us. They prepare our brains for the situation we're in. And we've also had, you know, if you had a really good day, students get a really good exam result, or you get a really exciting piece of data, that positive emotional experiences means that whole day feels better. And that's because we know that these biases influence our learning and memory and our decision making. So they have these quite widespread impacts, but also there's the potential to measure them. Because if we can design tasks, and this is what's been done in humans, we can design tasks which quantify these core kind of cognitive and executive functions, we can then look at how are those changed when somebody's in a negative affective state or a positive affective state, and then we can put a number on it. And I think that's really what we're all looking for, a readout, a quantifiable objective readout that we can use. And what we know from those studies in humans is major depressive disorders associated with negative biases, and there's a whole neuropsychological hypothesis developed around this idea, and it's very much supported by our work and also Catherine Harmer's work in Oxford. And it's quite interesting around this idea of delayed onset, because actually it suggests that Conventional antidepressants induce this acute positive shift in emotional processing. So the pharmacological effect of the drug is rapid and it changes the brain chemistry. But then you require these kind of social and environmental factors to interact with that biological state through things like learning and memory. And it's that learning and memory that then builds in a positive way to subsequently influence our subjective mood. And that takes a bit of time to happen. And utilizing the kind of background that we could glean from the human literature, we were able to develop affective bias assays for rodents. And these were particularly, and my particular interest was around studying antidepressant mechanisms. If we can understand better how the drugs we have work, then we can make better treatments potentially. So I've got two tasks. I'm going to briefly talk about judgment bias, but I'm going to talk more about affective bias test, mainly because the affective bias test was something we developed. The judgment bias tasks have been around for quite a while, actually. It was 2004 the first paper was published, and very much driven by the animal welfare literature, wanting, again, an objective way of understanding animal welfare. And these look at biases in decision-making associated with expectation of reward, avoidance of punishment, whereas the affective bias test is about reward, learning and memory. And this is an example here of our affective bias test, and this is a nice little operant rat. So the affective state-induced biases in decision making, this is based on this idea that in humans, if you are sort of glass half empty, glass half full, so optimism, pessimism idea. And when we're in a positive affective state, we tend to be more optimistic in the way we interpret our environment, the decisions we make, and the theory is animals would be the same. But how you do that, of course, is you first need to train animals to expect outcomes. So we would train our rats with a tone that predicts a high reward, a tone that predicts a low reward or avoidance of punishment. And once the animals learned that, we can test whether they're optimistic or pessimistic by playing them an intermediate ambiguous cue, looking at what they then, how do they anticipate the outcome of that? And we can see that by which lever that they press. And so this task actually is got some advantages and disadvantages. So it's operant, it's automated, so you can stick lots of rats into boxes, but it takes months to train them actually. It's surprisingly challenging. And we found lots of challenges actually around how they interpret the tone frequencies and all sorts of weird and wonderful things. But actually, overall, we did find depression-like models are associated with pessimistic behaviors and many others, and it's been seen in many species actually as well. Interestingly, we didn't find the same as in the humans, which was a shame. We found that only chronic treatment with antidepressants would shift to a more optimistic choice, not acute. And in fact, the acute pharmacology looked more dopaminergic, which is not really quite what we think we see in humans, possibly slightly different mechanisms then. Interestingly, the newer rapid-acting antidepressants, so drugs like ketamine, induced an immediate shift to a more optimistic, so that was really interesting. And we could link that to medial prefrontal cortex. So we had quite an interesting set of data. We had some evidence of construct validity. We had some evidence linking it to the right parts of the brain. We could differentiate between the different types of antidepressants. So perhaps the time course of antidepressant efficacy was picked up by the task. But we and others have looked at this. And so if you look at the sort of summary data, there's been several meta-analyses. And so what has proved to be more challenging is trying to understand what actually underlies the behavior we're seeing. Is it just a clever measure of motivation? A lot of the tasks that were used in animal welfare studies were more focused on a go, no go. So go for reward, don't go to avoid punishment. And that has a confound and that's posed an issue. And the pharmacology has not really been as consistent as we would have liked. And so you certainly do look at these meta-analyses to give you sort of a better idea of the overall picture. But from our perspective, we certainly think that there are benefits to using this in certain situations, particularly some of the evidence that animals in a more negative affective state will make more pessimistic decisions. So certainly from a phenotypic kind of assessment, but mechanistically and therefore then trying to identify novel drug targets, perhaps not quite so clear at the moment. This task is the one that we developed in our group. So this is the affective bias test, and it arose from an idea that we had that it wasn't sort of directly aligned to a human task. It was slightly to the side of that. So we knew that patients with depression have this very interesting when you talk to them, they attribute far less value to positive experiences than people who don't have depression. So they experience the same world, but they have this negative bias towards their kind of reward memories. So we hypothesize that perhaps the affective state at the time you're learning something positive can actually bias the memory that is then encoded for that experience. And then when you subsequently retrieve that memory, you'll see this kind of negative bias. So it will be less likely to drive behavior than under a control condition. And so we were lucky because, of course, we knew from condition place preference type protocols that you can teach animals to do these associative, independent associative learning under different conditions, and then you can probe their bias by using a preference test. Now, we knew from the literature that condition place preference is not sensitive to antidepressants, so we knew that we couldn't just use that approach. But what we designed was something that was much more about active learning. So rather than the sort of typical condition place preference protocol that's used for drug related studies and addiction related research, we had the animals in the same context, but we gave them 2 bowls, so a bit like this. And the 2 bowls, one of them has food in it and the other does not. And the animal is learning. So it's that we actually record trials to criterion. So they have to learn, they have to go to the correct bowl 6 consecutive times. Importantly, it allows us to pseudo-randomly spatially orientate the bowls, because one of the things we discovered very early on is you can't quantify affective biases based just on left-right choices. That just didn't work. And it's also why, at the end, I will tell you why we still haven't automated the task, which is disappointing, but is still a work in progress. So it's a manual bowl digging task. The animal goes in, they learn. One independent learning experience is that A has reward. The second one is B has reward. And we can either manipulate the amount of reward, so they can have 2 pellets and 1 pellet, which I'll talk about at that assay. We called that the reward learning assay. But in the affective bias test, which is the task we sort of looked at first, the actual reward value is kept the same, 1 pellet throughout. So the animals should have no bias. But we manipulate their affective state here before one of the learning experiences. We repeat it here. We have to repeat it because otherwise we get a recency effect. If we repeat this learning, we don't see a recency effect. The animals then genuinely use the substrate as their kind of predictor of reward value. And then we have our preference test. And if animals have been given a positive affective state manipulation here, we would expect to see a positive bias, so they make more choices of A. And if we give a negative manipulation here, we would expect them to make more choices for B. And from my main interest in pharmacology and antidepressants, we've looked at things that could modify new learning. So experiences that, as the animal is forming a new experience, can an affective state manipulation bias that. But also we were interested, particularly as we got into trying to understand the sort of conventional versus rapid acting antidepressant drugs, we wanted to know whether they interacted with these mechanisms differently, and I'll show you some of the data on that. And so what we do also is we can generate a negative affective state and a negative bias. So the bias is already there, and then we use the treatment before the retrieval part of it. So we acutely, or we can look at it 24 hours after the animals are given the drug, and then we look at their preference. So we're looking at, can you modify a new memory? Can you bias a new memory? And also, can you then modify a biased memory? And so here we have a guy doing the task. It's very easy at one level, but the caution I would say is that, as with any bowl digging task, you have this rather complicated, difficult to kind of quantify, as it were, interaction between the human and the animal. And so if I just play the video again, I think it will play again. The task relies on the handler giving the animals time to explore both bowls, and then knowing when the animal has made a choice. And that is quite difficult, actually. And you need the animal to be choosing one of the bowls, digging in it, and then you remove the other one. And that's how we know they've learned the task. But also, when we then come to quantifying the bias, we need the animals to be using and correctly reporting their preferences. So these are a whole load of data that several students' PhDs worth of data over the last sort of 10 years that we've done. And the first thing we did was proof of concept. So we just said, you know, if we give them 2 pellets on one day and 1 pellet, they should develop a bias, a preference towards the substrate that predicted the higher value reward. And that's exactly what we see. So we get a bias that's sort of between 10 and 15% in the population. All the rest of the data that you're looking at here, that's all actually been done using equal value rewards and dose responses. So all of these studies, apart from the psychosocial manipulations, all of these studies have a vehicle control that confirms 50-50, there's no bias. And then as the dose increases, you get a nice dose response. So a couple of things to highlight. Very exciting. The antidepressants all have an acute ability to induce a positive affective bias, and they're all at doses that are really quite clinically relevant. So unlike the forced swim test, where you would need 10 mg per kg or even 30 mg per kg fluoxetine, which is way over actually the occupancy of the target receptor, and it's not the same occupancy of receptor you see clinically. Ours are all working at kind of clinical doses, which is quite exciting for us, and really helps strengthen our belief that we're engaging relevant underlying mechanisms. We were also interested in what happens if we use the social manipulation, because affective state, as you know, affective state is not just about a drug, it's about, you know, social enrichment seemed like a really sensible way of, the animals should be in a more positive state. So we did this with the social arena. So we have a playpen, 8 rats go and play in the playpen, and during that day they're taken out, they do their learning experience, they go back in the playpen, and then that's compared with experiences learned when they're just in their home cage. And so you get a really nice positive bias. It's not as big as some of the drug studies, but it's very robust. We've seen it in male rats, female rats, and a couple of different strains, so it seems really reliable. Now, we were also really interested in, and actually the original funding came from a company who wanted to work with us to look at rimonabant really, and wasn't so interested in the positive, but interested in because the forced swim test sort of predicts these drugs. So, you know, we wanted to know whether or not where it failed had really been in some of these areas. So we did the pro-depressant side of it. And again, that's one of the, you know, reasons why we use the animal studies, because it wouldn't be ethical to do these studies necessarily in people. So here we've got drugs that we know have caused negative affective states in humans. And here we very clearly see these negative biases are generated by an acute dose. This was one of my most interesting pieces of data. I never knew rats really found restraint stress quite as aversive as it appears they do. So this is a 10-minute restraint stress. And this is almost as potent as this benzodiazepine inverse agonist, which in humans is pretty potently anxiogenic. And this has been a very robust finding. So restraint stress animals really does generate a potent negative affective state. I should also say that we see quite nice dose response. So it looks like that the level of the bias that is reported in the preference test does really closely reflect what was seen, what the animal experienced when they were learning. And you get a really quite tight dose response relationship. Giving them corticosteroid, that works as well, gives a negative bias, but also inflammation. So this, again, when I said at the beginning, you know, one of the challenges with the forced swim has been that it works for some areas, but not others. What's nice here is we seem to get this really consistent effect across loads of different drugs. And we've also got lots of drugs that are neutral, which I think is important as well. I haven't shown the data, but in the forced swim, again, people tend to always find an effect, whereas we actually find that a lot of drugs have no effect in this assay at all. Now, the interesting question, this one was that ketamine didn't work. And originally, I think we were a little disappointed. We thought, well, if ketamine's got this rapid acting antidepressant effect, that's a bit disappointing for our validation. And then we thought, well, hang on, it's a completely different drug. So why would we expect it to work in the same way as some of these other treatments, particularly as the positive biases were coming from these antidepressants that were, these were drugs which were conventional antidepressants. And so this led us to a different idea, and that was that perhaps we could use this test to explore whether or not the difference in the rate of onset of these drugs was linked to how they modulated these affective biases. And so we knew that ketamine didn't affect new learning, whereas the conventional antidepressants did. So we then thought, well, what about the other way around? What happens if we look at an established bias and then whether that can be manipulated? And so we had some really nice data showing that ketamine, the rapid acting antidepressant, but not venlafaxine, conventional antidepressant, can block this retrieval of the negative bias. And it also works with a stress-induced manipulation. And so we induce the negative bias, we treat with the compound, and we essentially lose the bias. It seems to take that bias away. And we've done the paper we published in 2015, as well as some of our unpublished data. So we've shown that there are very clearly different neuropsychological effects with delayed versus rapid acting antidepressants. And I think it really strengthens the kind of construct validity of the task, but also that drugs which work at very different time frames in humans also work very differently in our model. The Forswim rapid or conventional antidepressants, they all work the same. In the readout is the same. They all change in mobility time. So we also went to say, well, hang on, in people with affective biases and how they're processing affective biases, what we see is that the amygdala and the prefrontal cortex are really important. So we said, well, is our assay in rodents using the same brain circuits? And the answer is yes. And interestingly, perhaps differently. So conventional antidepressants seem to particularly target through the amygdala. So this is an amygdala lesion. And if you lesion the amygdala, you lose the positive bias to venlafaxine and you attenuate negative. So it looks like this is a really important area of the brain for biasing new learning. Ketamine doesn't, though. But if we look at the prefrontal cortex, here we see a different picture. So this is a negative bias. If we infuse the drug directly into the cortex, we see, here we go, ketamine, and you get this nice attenuation of the bias. And this is actually a 5-HT2A agonist. So this is a drug which is like the psychedelics, which of course there's a lot of excitement about how the psychedelics might be also antidepressant. And we think that perhaps part of their mechanism may be because they can modulate these biased memories, they can bring them to neutral. Now, of course, as any of you do behavior, just flip back actually, as any of you do behavior will know, one of the things as a behavioralist, when you see something go to 50-50, you can get very excited because you get a big significant attenuation, but equally, you could think, well, how do I know that this isn't just because the rat has forgotten who he is, what he is, how to do the task? Because of course, if he just doesn't remember how to do the task, if it's just this non-specific, then he would also give a 50-50 result. So we measure things like latency, we measure number of emissions to try to look for sort of more general impairments, but we also wanted to look at a memory, a similar memory, but something that wasn't induced by affective state. So we used our reward learning assay, which was quite nice. Here we've got this reward learning assay where we show that there's no effect at all. So we see that the ketamine can attenuate an affective state induced bias, but has no effect on a reward induced bias. And this is replicated with other types of antidepressants. So we think that there is a circuit in the brain that's an affective bias circuit that has important mechanistic role in how antidepressants work, possibly in causing depression as well. And it's rather selectively modulated by antidepressant drugs. And we've done other memory tests to show that these doses don't have any kind of general memory impairing effects. So I'm very conscious of time and it's taking me slightly longer. So I'm just going to very quickly mention the reward learning assay. And this is something we've been working on more recently, and it links into this idea that there's a lot of interest in rewarding activities and is a core symptom of depression. We've tried to model that in rodents by looking at things like sucrose preference, but that's not about this is all about experience of pleasure, not expectation. And we think it's expectation that's so important in depression. And in fact, the studies that have been done in human sort of sucrose preferences don't show impairments. So we needed another way of looking at reward related behaviors. And so we took our reward learning assay, this high reward, low reward, and we predicted that perhaps if they've got a reward learning deficit, when we run this assay in normal animals, we see this nice reward induced bias, but in depression models, that would be attenuated. And so these are data, and I'll just really highlight what you're looking at. So this is reward induced bias in our reward learning assay or a modified affective bias test, so just where we're using high versus low reward. And then this is sucrose preference test. So you can see here, the corticosterone looks like the literature. You see a deficit in sucrose preference. You also see an impairment in reward learning. But these are these other risk factors for depression. We know that prodepressant drug, we've got an immune challenge and early life adversity. These don't affect sucrose preference, but they do all affect reward learning. So we think this reward learning impairment could be a really good biomarker. We can certainly discuss it maybe when we do the questions. I get the feeling this could be something that we could look at in the home cage. If we could find a way of just very simply looking at animals' straightforward reward learning behaviors, perhaps this is something that, and our data would suggest that this correlates quite strongly with affective states, that animals in a poor affective state show this impairment in their basic reward learning. It's also quite interesting when we look at reward learning, we've got this impairment in maternal separated animals, so early life adversity. We also see a sensitization. So these animals are much more vulnerable to negative affective biases. So this is where we're starting to say, well, actually, maybe we've got some construct validity in this readout. So this may actually be an important construct that is driving depression. But again, nothing about reward sensitivity or motivation. So it's quite a specific reward learning deficit. We don't yet know a huge amount about the relationship between affective biases and reward learning, but we do have a correlation. So it suggests that they may well be overlap in terms of the circuits. And it's something that we're really keen to try and do a bit more exploration. This was a really small study, really. So quite surprising to see a correlation, but may not yet be a replicatable finding. And this is just to show that it's sensitive to antidepressants. So if we chronically treat with antidepressants, we can reverse this reward learning deficit. So a quite positive piece of data in terms of supporting the validity of that as a readout. Okay, so I'll just sort of summarize and give us time for some discussion, hopefully. So the reality, I think, is the force swim test and tail suspension test. So they measured stress coping behavior. They're very sensitive to changes in animals' experience of chronic stress, and they are certainly altered by quite a range of pharmacological treatments, and they do show changes in lots of different disease models. But exactly what they mean and how that relates to human depression is our real problem. Because we don't know how they relate, it's hard to then interpret the data, and there's this risk of kind of a misinterpretation or overinterpretation that a particular, you see a deficit in the force swim, therefore you conclude that this is a mechanism that's really important in depression. And there's tons of cell signaling molecules that have been targeted and shown to modulate this behavior. So sucrose preference test is similarly, you've got its challenges because it's reward sensitivity, but the reward deficits are slightly different in depression. And again, lots of the different depression models, it's really only sensitive to the stress model. And novelty suppressed feeding is probably more about anxiety. And again, it's not quite clear whether or not it's the same as what we see in the anxiety that happens in major depression. But of course, there's a lack of clinical biomarkers, so it's actually quite hard to establish translational validity. Affective biases represent this quite interesting. I'm interested in other people's thoughts. You know, should we actually be trying to develop cognitive biomarkers that are translational so that we can study these symptoms, these psychiatric symptoms across different species? And I hope I've given you some insight into how we've approached it. And I think at the moment, I'm quite confident the affective bias tests and reward learning assays, we've got more data than any of the other behavioral models have achieved in terms of construct predictive and face validity. The challenge is they're not high throughput assays. They're quite specialized. Training rats to do them is not as easy as it looks when you first see it. And we haven't yet managed to automate, and ideally we need automation. But I think what it is important is these affective bias tests may provide us with a ground truth. So we did this for our tickling study where we showed that ultrasonic vocalizations correlate with affective bias readout of affective state, which then means that we argue that we can now use 50 kilohertz vocalizations as a direct readout of affective state, because we've been able to show objectively and using this well-validated readout of animals' individual affective state that there is a relationship, a direct relationship, and therefore it's sort of, we could, you know, from the perspective of this group and the interest, perhaps this is something that could be used alongside home cage types of behaviors. So could we then use the affective bias test as a readout to then determine whether a particular set of behaviors that we're seeing in our home cage animals how does that relate to their affective state? And I think that could be really helpful going forward. And I just finish off by acknowledging lots of people who've worked on all of this. It's a huge project we've been doing for about the last 15 years with lots of collaborators. So thank you for your attention.